Have you ever come across while swimming in a lake or ocean and a green blob touches you and you begin to freak out completely? Can you believe that's the stuff that could replace non-renewable resources and could even stop global warming? Yes, I'm talking about algae. It's a completely renewable source and it's made from sunlight, carbon dioxide and water. Algae are basic food source for both salt and freshwater animals and also for birds. Their decomposition releases mineral into the water and their mass provide a refuge for small fish and invertebrates. If nature loves it so much, why don't humans? Microalgae have recently been researched as a molecular source for the manufacture of green materials. As microalgae generates polymer naturally, they may potentially be used in the development of bioplastics. Today, the use of various plastics is at an all-time high, almost all of which are oil-based. These oil-based plastics are derived from non-renewable resources which will ultimately become too costly to retrieve. So the demand for green materials have evolved to reduce the usage of these resources. A more sustainable life cycle for plastic goods could be produced by the use of these green materials. The purpose of this research is to develop a microalgae-based bioplastic material. In order to achieve this goal, extractions of polymers from the microalgae will be made. Extractions of polymer from plants can be difficult because they have multiple layered cell walls, making them tough to penetrate. In order to extract polymers without destroying their inherent structure, one has to damage the cell wall with a suitable method. In one of the studies, attempts to extract starch and PHA to create a bio-based plastic material were made by using these two different species. With the method washing with acetone and the use of ultrasonic homogenizer and dialysis, starch and PHA were likely extracted successfully. But due to a shortage of algae, very small amount of materials were extracted. In another study, chlorella species were used to extract PHA and were the most commonly used in the production of both biopolymers and plastic blends. Tests conducted to measure product quality showed that higher quality bioplastic could be obtained using chlorella species compared to spirulina species. However, spirulina species had better blending properties compared to chlorella species based on the physiochemical features of the material. Spirulina species, which has a higher production rate compared to chlorella species, will be used in my research to evaluate the efficacy of method used to extract PHA and to examine the influence of this method on the purity, properties and composition of polymer. So in my research, the procedure that will be followed is where the algae will be mixed with acetone for 24 hours at room temperature to remove pigments and excess lipids. 3 different analyses, which are SEM, FTIR and NMR, will be conducted to examine structural changes before and after the process and also to analyze what compounds were extracted. Following the washing procedure, the algae will be cryo-grinded to further damage the cell walls to achieve a higher extraction yield. The washed and cryo-grinded algae were mixed with 100 ml of sodium hypochlorite with a concentration of 12% for 1 hour at a room temperature. Water was added after 1 hour in order to precipitate the PHA since they are water insoluble. Solid and liquid phase were then separated using a centrifuge, where the solid phase, which theoretically should contain extraction PHA. All these approaches involved will help to evaluate many variables in this research. The method is considered to be one of the promising processes for the extraction of PHA from microalgae, and spirulina species will be used and proposed as the microalgae for PHA extraction in this study. So that's all from me. Thank you for listening.